Hi guys, and welcome to Chapter 10, Structure and Function of DNA. So really what we're going to be focusing on today is transcription and translation. So hopefully you guys can remember that from high school. If not, it is not as scary as it sounds. It's actually pretty easy, so we're going to work our way through it. Essentially all we're doing is inside the nucleus, we're going from our DNA, right? Remember that copy that I said that you have the architect who spent years and years trying to make this building, trying to make the blueprints. You don't want to lose that. You don't want to give it to your construction crew because they could lose it or damage it. Remember, your DNA is your only copy, your sole copy. So what we do is we make copies of that, right? Like Xerox copies of it, and that's what we send off to the construction workers, right? They're the ones who are going to read those blueprints, but they're all copies. They're not the master copy from the architect. And finally, the whole reason we're doing this is to build a protein. That's the whole reason. So based on your DNA, you build proteins. So this is known as the central dogma of biology. It's where we go from DNA to RNA to proteins. So that's what we're going to do right now. All right, so first off, this does come in two different steps. The first step is known as transcription, where you're making the copy. So remember, we had the architect, we had his big blueprints, right? To make a copy of that, you're Xeroxing, all you're doing is transcribing it. You're essentially writing it down. You're making a copy of it. Okay, so that's the transcription part of it. Later, the construction crew is going to read those blueprints, and they're going to translate that and turn it into a protein or a building. Okay, so the first step is where we're actually going from the DNA to the RNA, that is known as transcription. We're just making a copy of it. Essentially what we're going to do is we're going to open up that DNA double helix and we're going to read one side of it, because remember, the sides are complementary. A goes with T, C goes with G, always. So if we were to open one up, all you need to do is read the top half and you already know what the bottom half is going to be because they're complementary. So that's how RNA works. We open up the double helix, right, the double-sided DNA, we make one side copy, and that turns into our single-stranded RNA. So remember, DNA, double-stranded, RNA, single-stranded. And that's because, again, you really don't need both sides of the copy. You know, if you know one side, A, T, C, G, you know the other side's going to be T, A, G, C. That's it. Or whatever I said. All right, so that's, again, the two steps that we're going to be focusing on. Transcription, DNA to RNA, translation, RNA to the protein. And yes, I am actually going to have you guys making your own proteins, which is going to be pretty cool. Okay, so this is, again, exactly what that first picture showed you. We are inside the nucleus, protected where our DNA remains, right? This is our long, long strand of DNA, our double helix DNA. Through transcription, we turn that into a single strand of RNA. Now, if you notice, the RNA is already shorter than the DNA. That's because a lot of our DNA actually has what's called non-coding regions, meaning it doesn't really do anything for us. So why would we want to read this part of the DNA if it's not actually going to make us anything like our proteins? Okay, so we make a shorter copy of our RNA, and that's easier to read. It's easier to kind of, pick apart what are the good parts, what are the bad parts, what are the parts we don't need. So we can simplify our RNA from our super long strand of DNA to just a little bit of it. Imagine if you're the construction worker, you're just doing the bathroom, right? You're building that bathroom. You're not building the whole building. You don't need the instructions for the whole building. You're just building the bathroom. So you just have the instructions for that bathroom, okay? So the RNA gets made here inside the nucleus. Then the RNA is going to leave the safety of the nucleus to actually go make its protein, which is okay because our DNA still remains protected in that nucleus, right? But now our RNA, our single-stranded RNA, can come out of the nucleus safely, because what's the worst thing that's going to happen? Someone breaks our RNA, just make more. You have all the DNA you need. So if even when we're making this copy, if something goes wrong with the DNA, it's not that bad because you can always just make new RNA, right? So if you're reading the DNA and something gets happened and something funny gets made into maybe the wrong RNA, it's okay, you can scrap it and just make a new one because you have your master copy that's safe inside the nucleus. Okay, so transcription happens inside the nucleus, translation happens outside the nucleus in the cytoplasm. Remember the liquidy, just gooey part, things like where the glycolysis were happening? Same kind of thing here. Translation happens here in the cytoplasm. Okay, and that's where we're going from the RNA and turning it into an actual protein. Okay. So remember, we have these very long bits of DNA, but not all of them are what we need. So say this area right here is the coding area for your first gene, right? Remember, we already learned about genes. They carry our traits. We got these from our parents. Same kind of thing here. So this would be our first gene, non-coding region. Second gene, non-coding region. Third gene. Okay, so really we're going to break this down into genes, and those genes are going to produce our proteins, like the protein for curly hair, the protein for freckles, or a widow's peak, or any of the things that we talked about last time. Okay, so say this right here from here to here is the only part of the DNA that I need. Okay, so that's what we're going to actually make our RNA copy of. So essentially what we're going to do is we're going to open up that double helix. We're going to read the top half. That's going to be our DNA. And then finally, we're going to turn it into our RNA. So this is something that we're going to practice here in class. So the blue strand is your DNA. Through transcription, you're going to make a copy of it, right? Remember that in RNA, there are no T's, right? There's no thymine. There's only uracil. Okay, so A, instead of pairing up with T, A pairs up with U. Okay, so A pairs up with U, A pairs up with U, A pairs up with U. C always pairs with G, G always pairs with C, A always pairs with U, etc. Okay, so in this case, the way that I would do it is when you're writing out your RNA, because people get very confused about this. They go from A to U, but then if there's a T up here, they should go from T, right, to A. But they don't. They go from T to U as well. So now you have twice as many U's as you should. So what I always recommend doing is write out your original DNA strand is going to be given to you. Okay, I'm going to give that to you on a test. Just copy it into DNA. So in this case, it would be T, right? T, 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 G, G, C, C, G, T, 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 T. But because we're going to RNA, we know that there's no T, so then just cross off the T and go to a U. I know this is a little bit confusing. My example here doesn't have any T's in it, but imagine if this was T, T, T up here, right? T's don't match with U's, T's match with A's. There is A in RNA. So this would be, if this was T, 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 this would be A, not U, U, U. So make sure that you just practice that. And again, like I said, just write it out as if it was DNA, which we've already practiced, right? And then what you're going to do is just cross out all those little T's and make them into U's. Easiest way to do it. And if you have problems with that, practice it at home. If you guys want, write out a fake strand of DNA, copy it into what you think is the RNA, and send it to me if you want me to double check it. And I can tell you, yes, you got this wrong, or no, you got this right. Other way around. Yes, you got this right, or no, you got it wrong. All right, so now we have our RNA. Okay, so when you have the RNA, transcription is completed, right? You have your copy. Now what you have to do is you have to read that copy. You have to interpret that copy. That's the translation part. And so the first part of translation, what we do is we put things in what's called codons. And a codon is 3 nucleotides. So this would be a single codon right here. U, 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 that's a codon. That specific codon is going to code for a very specific amino acid. And remember, the whole point of this is to make a protein. And proteins are made of amino acids. We know that. We already learned that in macromolecules in chapter 4. Okay, so the whole point is to break these RNA down into little codons. Those codons are going to code for a very specific amino acid. Based on your sequence of amino acid is going to give you your protein. If this is confusing, don't worry about it. We're going to see all this over and over again today. Okay, so this picture kind of looks scary. I'm sure it looks scary on the video because we're so far away. But you can remember always zoom in on the PowerPoint and kind of follow through. It's really, really not that bad. So when we're actually going through the transcription part, remember the transcribing, we're just making a copy from DNA to RNA. What we're going to be doing is we have to start with the first step, and the first step is called initiation, right? It's not a shocker. You have to initiate. You have to start somewhere. So what the first guy is going to do who comes in is this guy called RNase polymerase. So you can see right here, RNA polymerase is going to make RNA. Anything polymerase you can think of as making or creating. So RNA polymerase makes RNA. DNA polymerase would just make more DNA. In fact, that's something that we need for mitosis, right? Mitosis and meiosis, when you're making copies of DNA, you're basically just going through, and it's the same exact steps except with a DNA polymerase instead of an RNA polymerase. So we're not making more DNA right now. We're not going through cellular replication. We've already done that. We don't care. Right now what we want to do is we want to make our proteins. So we need RNA to come in, read our DNA, so that we can actually start making our protein. So that's the first step of initiation. Okay? RNA polymerase is going to bind to the DNA. It's physically going to bind to the DNA, almost like sitting on top of the DNA. And it's going to bind specifically to the promoter area of the DNA. The promoter is kind of like, if you're trying to go somewhere, he's like, I have the directions. I know where I'm going, so come here first. Come to my house. We're all going to meet at my house, and then we're going to go off to the party or whatever we're doing. Okay, so RNA polymerase is like, hey guys, come here, start here. Okay, this is our starting point, because nobody knows where they're going. Nobody knows where the party is except the promoter. Okay, so the promoter is like RNA polymerase, come bind here. And really what he's doing is, remember all that non-coding region of the DNA? RNA polymerase doesn't know where to start. It's like, I don't know, is this my gene? Is this my gene? So the promoter goes, hey, this is the start of the gene that you're trying to make. This is the start of the protein that you want to make. Start here. Okay, so that's what the promoter does. He says, hey, come here, start here. So then RNA polymerase will bind to the DNA, right? And then it will actually be able to run down the DNA, opening it up and reading it. So that's exactly what's going to happen. So you can see here, initiation, the RNA polymerase is going to open up that DNA double helix from that tight wound spiral, right? It's going to break all those little hydrogen bonds that are connecting the A's and the T's and the C's and the G's. It's going to break all those little hydrogen bonds, open it up, and be able to read one side. Okay, so that's exactly what we see, well, not quite yet. All right, so step 1, initiation, right? It opens it up. Step 2, which I believe is here, step 2, this is actually where elongation begins. So step 2 is called elongation, which makes sense. 1, we initiated, we started. 2, now we're actually going to elongate our RNA. We're making our RNA strand based on our DNA. So that's exactly what's happening here. He's going to read all the way down this DNA, right? Opening it up and creating a growing strand of RNA. That's the whole point of this. Open up the DNA, read one side of it, make a copy of your RNA. And that's exactly what's going on right here. Now, step 3 is no shocker. It's called termination, right? You're done. So now we've actually read our DNA. We've gone through it. And now it gets to what's called a terminator sequence. Kind of like the promoter, where it's like, hey guys, start here. The terminator's like, okay, we're done. Like, our protein is complete. No need to keep going, right? That's not going to help us. Our protein is done. Well, our RNA is done. Our coding region is done, right? Our gene is done. Okay, this is the only part of the gene. Nothing else really matters after here. This is the gene that we were looking for. Okay, like the droids that you were looking for, these are the genes that you're looking for. So the promoter's like, hey guys, they're right here, and the terminator's like, okay, this is our endpoint. So that's it. Step 1, initiation, open up, right? RNA polymerase binds to the DNA, opens it up, starts reading it. As it reads it, it starts copying down RNA, that's the elongation part. And then finally it gets to the terminator sequence, and it goes, okay, we're done. This is our completed RNA sequence. Okay? So, kind of close-up of what's happening here. We are in our RNA polymerase right here. We've opened up our DNA. Remember, blue is DNA. And all we're doing is we're reading the bottom half right here, and we're putting these codons on to match. T goes with A, T goes with A, G goes with C, G goes with C, A goes with U. Okay, so if you want to look in here, you can kind of see that a little better than the last slide that we saw. So this is all it's doing. It's working its way through the DNA, and as it does, it's laying down each one of those nucleotides depending on its corresponding complement based on the DNA. Okay, once it's done, we have a nice long strand of this RNA right here, and then we reach the terminator sequence right here, and then it goes, the RNA no longer binds to the DNA, it leaves the DNA, right? And now you have your finished RNA, which is right there. Okay, so the next part that has to happen, remember we've just made a long strand of RNA. We're still in the nucleus, right? And our RNA isn't quite finished. Yes, we made a copy of it from the DNA, but it's not quite perfect yet. So what we have to do is what's called RNA processing. And essentially, it's kind of think of it as like meat processing, although better for you because processed meats aren't necessarily good. Okay, but what we have here is in our long strand of DNA, there are places that are called exons and introns. And only the exons are the good part of the DNA. The exons are the thing that get expressed. Like curly hair, that's an expression. Freckles, that's an expression of your gene. It's an expression of your DNA. Okay, so if you have freckles, you have the gene, and that expresses those freckles. Okay, so the exons are the good ones that express the, your physical traits. The introns are kind of in between. They don't really do anything. Okay, so to do RNA processing, we now have read a full copy of both the exons and the introns from our DNA, but we don't want the intron part. Okay, so what we have here, the dark parts are the good DNA, RNA, the light parts are those introns, right? We don't need those. So we do splicing. Essentially, we take out all the introns and we essentially just cut them out and throw them away. So we cut here, cut here, we get rid of these little fractions, and now we have just the coding region of the RNA, the good part. This part expresses this gene and this gene and this gene and this gene. There's no in between, right? There's no nonsense like these introns that don't do anything. Why would we have that? That's energy wasted. So we just get rid of those. We get rid of the introns, we keep the exons. Now, what we also do is we stick a cap, what's called a cap, on the front end of it. This cap is also kind of like the promoter, but for the ribosomes. Remember that ribosomes make proteins. So this is where that mRNA is going. It's going to a protein because it is the instructions for the ribosome. The ribosome is going to read the RNA and then be like, cool, take this amino acid, put it here. Take this amino acid, put it here. Take this amino acid, put it here. And that's how you're going to get your growing amino acid chain, which is going to turn into your protein. So until then, we have to make sure that our RNA is packaged nice and tight. So we stick a cap on the first end. This is kind of like the driver. He's like, I know how to get to the protein, or I know how to get to the ribosome. Okay, this little capper here also helps to bind to the ribosome so that the ribosome can actually read the RNA. We're going to learn about this in just a second, but you don't want to start on the wrong letter. If you're supposed to start on A, you need to start on A. If you start on a different letter, things can get messed up. So this cap kind of acts as like a protective little beginning that helps bind to the ribosome and say, start here. Start making your protein here, not here, here. Okay, so that's what the cap does. Now the tail is kind of same kind of thing. We have the tail over here. And this tail basically is a series of repeating A's. So it's just A, really long chain of A's. And that's because we want to make sure that we got all of our protein. When we're at the end and we're reading codon after codon after codon after codon, we want to make sure that we get the very last codon because then our protein is going to be incomplete, right? That's not what we want. So we just put a bunch of A's at the end and we go, cool. Now once we get to that repeating A section, we know, oh, okay, our protein's done. We're good. And then you can just break off that tail. Okay, so the cap and the tail are kind of help to package all of these RNAs so that one, you have a place to bind to the ribosome, and two, your protein doesn't get like lost, like the last end of it doesn't get lost or anything like that. You want to make sure that you get all of that protein. So once you're done with the mRNA, I say mRNA because there's a couple different types of RNA we're going to learn about. mRNA is messenger RNA, and that's what this guy is. He's sending a message to the ribosome. Here's how you build the protein. Here's the protein that we need. So that's why he's mRNA. He's a messenger RNA. So if you hear me say mRNA, because it's coming out of the transcription part, that's what I'm talking about, the messenger RNA, who's going to the ribosome with a message. Here's the protein. Please make my protein for me. Now, here's a common misconception that people have a lot of the times when they're learning this stuff. They think that the mRNA turns into a protein. That's impossible. This is made up of nucleotides. A protein is made up of amino acids, right? Remember we learned macromolecules? Nucleic acids are different than proteins, right? So the proteins are all made out of amino acids, which are naturally found in your body. The ribosome just grabs them and sticks them together and builds a long chain of them. The RNA does not turn into a protein. This is a common misconception that people get wrong every semester. All right, so once you have our packaged, processed RNA, what we're going to do is we're going to send it outside the nucleus. We have everything completed. Now let's send it outside the nucleus to our ribosomes to actually make our protein. So that's what you see right here. You actually see the little RNA strand come out of the nucleus. Okay, so just a bigger picture of what was going on here. We have exons and introns. Remember, exons express, those are the good ones. Introns are kind of in between. We don't need those. So we do mRNA processing. We stick a cap on the first end because that's what's going to bind to our ribosome to get this whole process started. We put a protective tail on the end so that nothing breaks off. We don't lose the back half of our protein. and then we splice out the introns, keeping the exons so that now we have a smaller, more concise, just coding region of our RNA. So then we can send that outside the nucleus as our finished package instructions, or mRNA, messenger RNA. Okay, so imagine this is my protein right here, right? Big and long and complicated. But what's actually happening is we're building these by codons. Remember, codon is just a sequence of 3. So this guy is known as a tRNA. Okay, tRNA is transfer RNA. All he's doing is bringing in those amino acids. Remember how I told you that the ribosome grabs amino acids and sticks them in a chain? They're brought in by tRNA. Okay, so tRNA is just like it's like one of those enzymes that's picking it up, taking it where it needs to go, dropping it off. Okay, so the tRNA is going to get recycled. It's in this case picking up an amino acid, taking it where it needs to go to the ribosome, and then dropping it off, and then it'll get recycled. The important part about tRNA is they're very specific for amino acids. They can only pick up one type of amino acid. And what type of amino acid they can pick up is based on their shape. Okay, so they have to find a complementary shape, which is called an anticodon. Okay, so an anticodon would be the opposite of your codon. Say your codon is GGG, okay, just to be simple. GGG is your codon, right? That's what you read from your RNA. Your anticodon would be the opposite of that. What fits GGG? CCC. So in this case, this would be CCC. And that would fit perfectly in with the codon of the RNA in the ribosome, which we're going to see right now. Now, you do not need to know absolutely all of these subunits. You don't need to know the P site and the A site and all these different large subunits and small subunits. You really don't. I would probably have you guys do this if we were actually meeting in class. But just to simplify things, don't worry about that this time. So just know that these are just binding regions where the tRNA is going to come and sit. So the tRNA is going to come bind to the ribosome, okay? And that's where it's actually going to start sticking on these amino acids. So what we have right here, come here, camera. All right, what we have right here is we have our cap, right? We have our what's called a start codon, and nothing can start until you actually have that start codon, which we're going to talk about. Now we have our actual coding region of our mRNA here. So what happens is it binds, right? So this little cap is going to bind in here, and then it's going to get fed through the mRNA binding site, and it's going to get read. And as it gets read, it's about boop, 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 boop. As each one of these goes by, boop, 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 it's going to read the codons. Codon is a sequence of 3. 1, 2, 3. It's going to find the complementary tRNA that fits that codon, right? So the tRNA is going to have the anticodon that fits the codon. Okay, it's the only one that can fit, so the right RNA has to come in, and it's the only one that can bind here because it's the only one that can fit, just like our enzymes. And it's going to bring a specific amino acid with it. Okay, so say this guy is going to drop off his amino acid here, and then he takes off. Next one's going to come in and bind, drop off his amino acid, then take off. So this chain of amino acids is going to get longer and longer and longer based on all these codons. Oops. Okay, so just like transcription, translation has 3 steps. And oh my god, they're the exact same 3 steps. Initiation, you have to start it off. Elongation, you're reading the mRNA this time, and you're elongating a protein. And then finally, termination. You get to your stop sequence, and then your ribosome knows, oh, we're done. I've made my amino acid, we're good. Okay, so 3 steps, same 3 steps as transcription. Okay, so initiation starts it off. The mRNA binds to the ribosome, that little cap, that little protective cap, goes in first, because it's the thing that is going to bind to the ribosome and get all this started. Okay, and then what's going to happen is you're going to bring in your first amino acid with your first tRNA, transfer RNA. It's bringing in the amino acid. This is going to get everything started. Now we have the first amino acid. Now we can tack on all those amino acids afterwards. Okay, and again, it binds at the cap, which I already think about, yeah. Okay, so This is our start codon. We're going to talk about that in just a second, but there is only one start codon, so you have to have that start codon. Nothing will happen until this little guy reads the start codon. Then, this little initiator tRNA, because he fits the start codon, his anticodon is exactly complementary to the start codon, so he's the only one that can come in. In this case, it is the amino acid methionine. So methionine is the first one. It's going to sit here with our little initiator RNA, and he goes, okay guys, we're ready. Let's start making a protein. Okay? So my start codon is AUG. Yes, you will need to know that. AUG is the start codon. The anticodon is UAC. You don't need to know that. But this is just the start codon. It's basically just going to come anticodon. It's going to fit there. It's going to bring in methionine, and now you're actually going to have your the start of your amino acids. So this guy basically sits right here, and now he's waiting for the next tRNA to come in. He's going to take off, but their amino acids are going to attach, and then he's going to move from here to here, and then he's going to take off. So essentially, you come in here, and then you come here, and then you take off. Okay, so that's what we see right here. He's going to come in here, he's going to attach his string of amino acids, and then he's going to take off. Okay, so he comes in, based on the codon, his anticodon fits as a complement, right? Then he's going to attach his amino acid, actually technically the P site attaches the amino acid to the A site, and then the P site takes off. This guy moves over one, now he's in the P site, and again, another tRNA is going to come in, sit here, drop off its amino acid to him, he's going to take off, this guy's going to take off, which is what you see right here, he takes off, the amino acid chain is getting longer and longer and longer, he moves over one, and then we repeat the process again. So it's really, this picture looks a lot scarier than it is, but it's really not. The codon gets recognized, you bring in the appropriate anticodon via the tRNA, because the tRNA is bringing, the tRNA has the anticodon, but it's bringing in that amino acid that we want. So we have to wait for this guy to come in here, bind perfectly, right? He's going to throw his amino acid on this chain, which is what we see right here. This guy is then going to take off to go get another amino acid. He's going to move over, shift, and then we're going to have another guy come in. And again, this is the elongation process. One comes in, attaches his amino acid together, he leaves. This guy moves over, another one comes in, attaches an amino acid, this guy leaves. Right? I'm going to show you guys a video of this. Hopefully it'll be a little bit easier to understand than just up here. But can you imagine if I was trying to do this without this diagram? It's impossible. All right. Okay. So that's what we have right here. Now, when we're done, we get to what's called the stop codon. Now, there are several stop codons. Even though there's only one actual start codon, there are several stop codons. You do not need to know what they are. You do not need to memorize them. You just need to know when you reach a stop codon, you stop. Yes, there's going to be codons after that stop codon, but you don't read them, right? Just like anything before the start codon, you don't read. You haven't started yet. The start codon, nothing can happen until the start codon comes in, that AUG. Then you're going to elongate, and then when you reach that one of the stop codons, then you have to stop. So even if there's stuff after that, you have to stop. Well, you have to stop because it's part of your test, but your proteins will naturally stop because that's their stop codon. That's where they'll stop. Okay. So, complete overview of transcription and translation. We are inside the nucleus. We take our DNA. It gets opened up by RNA polymerase. RNA polymerase is going to open this DNA double helix. It's going to read one half of the DNA. It's going to make a complementary strand of RNA. Right? It's got, this RNA now has to go through processing. It's got introns and exons. We want to keep the exons, get rid of the introns, stick a little cap on it, stick a little tail on it. Remember, the cap is for binding to the ribosome. The tail is kind of like a protective little end. Right? Then once we're done with RNA processing, we're going to leave the nucleus. We're going to join up with our good friends tRNA and, where are we? tRNA right here and the ribosomes. Don't worry about the enzymes. tRNA and the ribosomes, they're going to bind based on the anticodons, right? They're complementary anticodons. Once you have the start codon, AUG, you're going to get red. You're going to elongate. You reach the termination codon, stop. Okay, and that's what we see right here. So we've initiated We're elongating, making it longer and longer and longer. Finally, we reach termination, right? The tRNA leaves, and now we just have a final protein, right? Amino acid string, which is just a protein. See, that wasn't so scary, right? Hopefully it wasn't. If you have any questions, again, watch the video again. I also have a little animated video for you guys to see right now.
Okay, roll it. You know what this is? It is the longest word in the world. Like anywhere, any language, more than 189,000 letters. If you were to write it down, though I don't know why you would, it would fill up more than 100 pages. And if you could actually say it without like breaking your face, it'd take about 5 hours. What the frick is this word? Well, it is the name of the longest known protein on Earth, and it's actually in you right now. Because of its enormous size, it was given the name Titan by scientists, and that's with 2 I's. And it's a protein that helps give some like the springiness to your muscles. Today we're going to be talking about DNA and how it, along with 3 versions of its cousin RNA, unleash chemical kung fu to synthesize proteins just like this. So this is going to take a while to explain. So how about we make ourselves some Hot Pockets? Oh, my favorite. Ham and cheese. Every time I take a bite, I wonder, how do they do it? How do they pack exactly the same flavor into every foil cardboard wrapped food-ish item? Clearly there's got to be some super secret instruction manual kept and a location known only to 2 people. And since I'm talking about biology here, that brings up a related question. How did I get built from the DNA instructions and biological molecules we've been talking about? Today, that's what I'm going to do, not actually make Hot Pockets or a person, but I'm going to be talking about DNA transcription and translation, which is how we get made into the delicious things that we are today, though hopefully none of us know how delicious people are. Animals, plants, and also Hot Pockets really are nothing more than salty water, carbohydrates, fats, and proteins combined in precise proportions following very explicit instructions. Let's say I want to make my own Hot Pocket. I would have to, 1, break into the lair of the Hot Pocket company holding the secret manual. 2, read the instructions on how to make the machinery to produce the Hot Pocket and the proportions of the ingredients. 3, quickly write down that information in shorthand before I get caught by the Hot Pocket police. 4, go home, follow the instructions, build the machinery, and mix the ingredients together until I have a perfect Hot Pocket. And that is how we get us. Very simply, inside the cell's nucleus, the DNA instruction manual is copied gene by transcription onto a kind of RNA and then taken out of the layer where the instructions are followed by the process of translation to assemble amino acid strings into polypeptides or proteins that make up all kinds of stuff from this Titan down here to the keratin in my hair. But most of the polypeptides that get made aren't structural proteins like hair, they're enzymes which go on to act like the assembly machinery, breaking down and building and combining carbohydrates and lipids and proteins that make up variations of cell material. So enzymes are just like whatever ingenious machinery they use in the factory to make this. Okay, let's start out in the layer, the nucleus. The length of DNA that we're going to be transcribing onto an RNA molecule is called our transcription unit. And let's say, in today's example, that it's going to include the gene that transcribes for our friend Titan, which in humans, at least, occurs on chromosome 2. Now, each transcription unit has a sequence just above it on the strand, and that's called upstream. Biologists call that upstream on the strand, and that sequence sort of defines when the transcription unit is going to begin. This special sequence is the promoter, and it almost always contains a sequence of 2 of the 4 nitrogenous bases that we talked about in our last episode: adenine, thymine, cytosine, and guanine. Specifically, the promoter is a really simple repetition. We got thymine, adenine, thymine, adenine, and then A, and then on the other side, A, T. Because you know how this works, right? This is called the ta-ta box. It's nearly universal, and it helps our enzyme figure out where to bind to the strand. Now you'll remember from our episode about DNA structure that DNA strands run in one of 2 directions, depending on which end of the strand is free and which end has a phosphate bond. One direction is 5' to 3', and the other direction is 3' to 5'. In this case, upstream means toward the 3' end, and downstream means toward the 5' end. So the first enzyme in this process is RNA polymerase, and it copies the DNA sequence downstream of the ta-ta box, that's towards the 5' end, and copies it into a similar type of language messenger RNA. Quick aside, so you'll notice that to read the DNA in order to make enzymes, we need an enzyme in the first place. So it kind of gets chicken egg here. We need the enzyme to make the DNA and the DNA to make the enzyme. So where did RNA polymerase come from in the first place if we haven't made it yet? What an excellent question. It turns out that all these basic necessities get handed down from your mom. She packed quite a lot more into her egg than just her DNA, so, we had a healthy start. So, thanks, Mom. So the RNA polymerase binds to the DNA at that ta-ta box and begins to unzip the double helix. Working along the DNA chain, the enzyme reads the nitrogenous bases, those are the letters, and helps the RNA version of those nitrogenous bases floating around in the nucleus to find their match. Now, you might also recall from our previous episodes that nitrogenous bases only have one counterpart that they can bond with. But RNA, which is the pink one here, doesn't have thymine like DNA does, which is the green and the blue. Instead, it has uracil. So U appears here in T's place as a partner to adenine. As it moves, the RNA polymerase rezips the DNA behind it and lets our new strand of messenger RNA peel away. Eventually, the RNA polymerase reaches another sequence downstream called a termination signal that triggers it to pull off. Now, some finishing touches before this info can safely leave the lair. First, a special type of guanine is added to the 5' end. That's the first part of the mRNA that we copied, and that's called the 5' cap. On the other end, it looks like I fell asleep with my finger on the A key of my keyboard, but another enzyme added about 250 adenines onto the 3' end. This is called our poly A tail. These caps on either end of the mRNA package make it easier for the mRNA to leave the nucleus. They also help protect it from degradation from nearby passing enzymes, while also making it easier to connect with other organelles later on. But that's still not the end of it. As if to try to confuse me to protect the secret Hot Pocket recipe, the original recipe book also contains lots of extra misleading information. So just before leaving the nucleus, that extra information gets cut out of the RNA in a process called RNA splicing. And it's something like editing a video. This process is really complicated, but I just have to tell you about two of the key players because they have such cool names. One, the SNRPs, which are small nuclear ribonucleoproteins. These are a combination of RNA and proteins, and they recognize the sequences that signal the start and end of the areas to be spliced. SNRPs bunch together with a bunch of other proteins to form the spliceosome, which is what does the actual editing, as it were, breaking the junk segments down so their nitrogenous bases can be reused in DNA or RNA, and sticking together the two ends of the good stuff. That good stuff that gets spliced together, by the way, are called the exons because they will eventually be expressed. The junk that gets cut out are just the intervening segments or the introns. The material in the introns will stay in the nucleus and get recycled. So, for instance, Titan down there is thought to have hundreds of exons when it's all said and done, probably more than 360, which may be more than any other protein. And it also contains the longest intron in humans, some 17,000 base pairs long. Man, Titan, it is just a world record holder. So now that it's been protected and refined, the messenger RNA can now move out of the nucleus. Okay, so a quick review of our Hot Pocket Mission Impossible caper so far. We broke into the layer containing the instructions, we copied down those instructions in shorthand, we added some protective coatings, and then we cut out some extra notes that we didn't need, and then we escaped back out of the layer. Now I have to actually read the notes, make the machinery, and assemble the ingredients. This process is called translation. So next, rewind your memory or just watch that video again to the episode about animal cells. Do you remember the rough endoplasmic reticulum? I hope you do. Those little dots on the membranes are the ribosomes. And the processed messenger RNA gets fed into a ribosome like a dollar bill into a vending machine. Ribosomes are a mixture of protein and a second kind of RNA called ribosomal RNA, or rRNA. And they act together as a sort of workspace. rRNA doesn't contribute any genetic information to the process. Instead, it has binding sites that allow the incoming mRNA to interact with another special type of RNA, the third in this caper, called transfer RNA, or tRNA. And tRNA really might as well be called translation RNA because that's what it does. It translates from the language of nucleotides into the language of amino acids and proteins. On one end of the tRNA is an amino acid. On the other end is a specific sequence of 3 nitrogenous bases. These two ends are kind of matched to each other. Each of the 20 amino acids that we have in our body has its own sequence at the end. So if the tRNA has the amino acid methionine on one end, for instance, it can have UAC as the nucleotide sequence on the other end. Now it's just like building a puzzle. The mRNA slides through the ribosome reads the mRNA 3 letters at a time, each set called a triplet codon. The ribosome then finds the matching piece of the puzzle, a tRNA with 3 bases that will pair with the codon sequence. That end of the tRNA, by the way, is called the anticodon. Sorry for all the terminology. You need to know it! And of course, by bringing in the matching tRNA, the ribosome is also bringing in whatever amino acid is on that tRNA. Okay, so starting at the 5' end of the mRNA that's fed into the ribosome, after the 5' cap, for almost every gene, you find the nucleotide sequence AUG on the mRNA. The ribosome finds a tRNA with the anticodon UAC, and on the other end of that tRNA is methionine. The mRNA, like a mile-long dollar bill, keeps sliding into the ribosome so that the next codon can be read, and another tRNA molecule with the right anticodon binds on. If the codon is UUA, then the matching tRNA has an AAU on one end and a leucine on the other. And if the mRNA has an AGA, then the matching tRNA has a UCU on one end and an arginine on the other. In each case, that new amino acid gets connected onto the previous amino acid, starting a polypeptide J, which is the beginning, very beginning of a protein. But it turns out that there are lots of different ways to read this code, because UUA is not the only triplet that codes for leucine. UUG does too, and arginine is coded for by 6 different triplets. This is actually a good thing. It means we can make a few errors in copying and transcribing and translating DNA, and we won't necessarily change the end product. The process continues with the mRNA sliding in a bit more, and the ribosome bringing in another tRNA with another amino acid, and that amino acid binding to the existing chain, and on and on and on and on and on. Sometimes for thousands of amino acids to make a single polypeptide chain, for example. This whole word is basically just the names of the amino acids in the sequence and the order in which they occur in the protein, all 34,350 of them. But before we can make our own hot pockets, and that string of amino acids becomes my muscle tissue, we have some folding to do. That's because proteins, in addition to being hella big, can also contort into very complex and downright lovely formations. One key to understanding how a protein works is to understand how it folds. Scientists have been working for decades on computer programs to try and figure out protein folding. Now, the actual sequence of amino acids in a polypeptide, what you see scrolling along down there, is called its primary structure. One amino acid covalently bonded to another, and that one to another, and to another in a single file. But some amino acids don't like to just hold hands with 2 others. They're a bit more promiscuous than that. The hydrogens on the main backbone of the amino acids like to sometimes form bonds on the side, hydrogen bonds, to the oxygens on amino acids a few doors down. When they do that, depending on the primary structure, they bend and fold and twist into a chain of spirals called a helix. We sometimes also find several kink strands laying parallel to one another called pleated sheets. All those hydrogen bonds and pleated sheets are what makes silk strong, for instance. So in the end, our promiscuous amino acids lead to wrinkled sheets. These hydrogen bonds are what help give these polypeptides their secondary structure. But it doesn't end there. Remember the R groups that define each amino acid? Well, some of them are hydrophobic. And since the protein is in the cell, which is mostly water, all of those hydrophobic groups try to hide from the water by huddling together. And that can bend up the chain some more. Other R groups are hydrophilic, which, if nothing else, means they like to form hydrogen bonds with other hydrophilic R groups. So we get more bonding and more bending, and our single file line has now taken on a massively complex 3-dimensional shape. It also explains why I can fix my bed head by wetting my hair with water. The water helps break some of those hydrogen bonds in the keratin, which relaxes its structure. That way I can comb it out, and when it dries, those bonds reform, and voila, perfect hair. All of this shape caused by bonding between R groups gives our polypeptide its tertiary structure. So now we have a massively contorted polypeptide chain, and it actually contorts very precisely. Sometimes just one chain is what makes up the whole enzyme or protein. In other proteins, like hemoglobin, several different chains come together to form a quaternary structure. So quick review of structure. The sequence is the primary structure, the backbone of hydrogen bonds forming sheets and spirals for the secondary structure. R group bonds are tertiary in the arrangement of multiple proteins together, give the quaternary structure. These polypeptides are either structural proteins, like this thing at the bottom here, that you can find in your muscle or in my hot pocket. They might also be enzymes. Enzymes like do stuff. They can cut up biological molecules like I do with the chef's knife. They can mix stuff, and they can put stuff together. So from that one recipe book, we get all the ingredients and all the tools necessary to make me, which is better than a hot pocket. Would you all agree? Now take your time with this stuff. Feel free to watch the episode a couple of times, because next week we're going to talk about how cells swap all of this genetic information through reproduction. Thank you for watching this episode. By now, you should probably know how this works. You can click on any of the links over there, and it'll take you back to that point in the show, as long as you're not watching on your cell phone. It doesn't work on cell phones. I apologize for that. Thank you to everyone who helped us put this show together, and thank you to you for watching it today. If you have any questions about this episode, please leave them in the comments below, or you can get us on Facebook or Twitter. That's all. Goodbye.
Okay, so hopefully that helped you kind of understand, at least get a visualization of what was happening during transcription and translation. If you want to, you can always rewatch his videos again. It's just crash course. He's got all sorts of information for you. It's really, really great. If he speaks a little bit too fast or you don't really like his videos, remember you guys have Google at your fingertips, okay? We're all stuck at home now, so you might as well go through and actually Google this information. You can see a million different videos on it, different animations, different people explaining it. Hopefully one of them will be able to help you. If not, that's what I'm here for, and you can always email me. All right, so we talked about making our proteins, right? Going from DNA to RNA to an actual protein. Well, something that he already mentioned in the video is, well, what if we make a mistake? Well, luckily, you know, a lot of those sequences, a lot of those codons will code for multiple amino acids, meaning there's like 6 different codon sequences that will code for arginine. So if you mess one of those up, it's still arginine, so your protein will end up the same way. Sometimes that happens in the copying process. Sometimes it happens via what's called a mutation. Now, this is not Wolverine mutations and you're going to turn into the Hulk. That's not how that works. But these mutations are actually pretty good sometimes. Sometimes they're bad, but sometimes they lead to things like evolution because they lead to adaptations. There's variation in the population. That variation will lead to evolution, which we're going to talk about in the next couple weeks. So a mutation is basically any change in your original DNA sequence. Mutations can actually affect the amino acid, meaning if you have an A that turns into a G, now you maybe have an entirely different amino acid, so your protein is going to be different. Sometimes that A goes to a C, and there is no difference. So we're going to learn about the different types of mutations coming up in just a second. So mutations can either be large mutations, or they can be very, very small mutations, right? Larger mutations, you can imagine, are going to cause bigger problems. Smaller mutations, meaning that A just went to a C, is probably going to only affect one single amino acid in your very long protein chain, right? So that mutation probably won't be such a big deal. However, we've already learned about our good friend sickle cell, who is a single, what's called a point mutation, meaning a single nucleotide has been changed. So in this case here, we have our normal DNA, we have our normal RNA. Right? We produce this normal protein where we have glutamine right here, and we produce normal red blood cells. So if we do have that point mutation, remember, a single nucleotide is being changed. We're supposed to have a T in this position, but based on our mutation, we now have an A in this position. So instead of going from a T to an A when it comes to our RNA, we go from an A to a U. The presence of this U right here actually changes the amino acid from glutamine to valine. Okay, this is a different amino acid. This is the wrong amino acid. Now the protein is going to fold in a slightly different way, like we just learned about, and now we have a sickle cell red blood cell, right? That curved, almost crescent moon shape, instead of our nice big healthy round blood cell. So in this case, a single nucleotide mutation from that T to an A gives you sickle cell. But what are you not going to get if you have sickle cell? That's right, malaria, right? We learned about that last week with genetics as well. Okay, so in this case, it's not going to kill you, but it is going to affect you for the rest of your life, and because it is a mutation in the DNA, your kids are going to get this too, because they're going to get your DNA. So, kind of sucks. All right, so here are the different types of mutations. We have what's called a nucleotide substitution. You are simply substituting one nucleotide for another. That would be the one we just saw. An A to a C, a T to a G. Whatever you're substituting, you're just substituting one. Now, these nucleotide substitutions are usually point mutations, meaning a single nucleotide has been switched. Okay, that's a single point, has mutated. But sometimes that single point mutation can actually be really bad if you have what's called a deletion or an insertion. Remember, you're reading things based on the reading codon. It's called the reading frame, meaning you start at the first word, then you go to the second, the third, and fourth, and fifth. What happens if you lose the fourth word? That sentence doesn't make any sense anymore. Same kind of thing here with your DNA. So if you're supposed to be a sequence, and I'm going to do this very, very simple sequence on the board. Okay, so I have T-A-G-A-G-C. Okay, that is my sequence. Say it's coding for a protein, right? This would be my first codon, T-A-G. Okay, my second codon would be this one here, A, G, C. Okay? If I had a single point mutation and it's a substitution, maybe this goes from TAG to TAC. Not that big a deal. The rest of it, as it goes on, is all going to remain the same. But what if I completely delete this C? Now instead of having, so say this guy got deleted here. So instead of having TAG and then AGC, now I have TAA. So now my reading frame is what's called shifts. This is called a frame shift mutation. Insertions and deletions cause frame shift mutations. It means every single codon after that is going to be wrong. Because now I have TAA, GC, maybe it takes over from the next codon. It shifts the entire thing. Every single codon is going to be wrong afterwards, which means every single amino acid is going to be wrong after this, which means the whole protein is not going to work. Right? This is where things get bad. So these are the bad types of mutations. Okay? Substitutions, not so bad. One single, nucleotide got changed. Maybe it changes one amino acid. Maybe it doesn't. Okay? But a deletion or insertion, these are really bad. Right? These cause what's called frame shift mutations. Okay? So these are bad because it changes the reading frame of the entire genetic code. Now everything is one letter off, which means everyone's going to get messed up. All of your instructions, essentially, are messed up, which could be some really, really bad stuff here. So that's what we see right here. We have a single point mutation and a single substitution. Okay, so all of these are point mutations, but the substitution is the only one that's not a big deal. Okay, so we have methionine and lysine and phenylene and serine and alanine. Right? So now we went from a G, in a single substitution, we went from a G to an A. Well, the only one that's going to get affected is the serine right here. Okay, maybe he was supposed to be glutamine, or what is this, glycine? Glycine. It's supposed to be glycine, but now it's serine. Okay, but every single one after that is going to remain the same. It's just a single substitution. That's not so bad. However, when we have the nucleotide deletion, that's when things get bad. So now we're supposed to be UUG, right? And it's supposed to be a leucine. Now we delete it, right? It's probably supposed to be UUU. We deleted it, now it's UUG. So everything gets shifted afterwards, which means leucine is wrong, alanine is wrong, whatever after here is wrong. They're all wrong because we have the deletion. Same thing right here, we have an insertion. Okay, so now it's supposed to be UUU, but we've inserted a G. Okay, so now it's UUG. Again, we're at the wrong amino acid. Wrong amino acid. Wrong amino acid. It shifts the entire thing, causing all the codons to be wrong, which causes all of the amino acids to be wrong, causing your protein to fold all weird and funky. And probably not what it's supposed to be because you had a code, you had the instructions. Essentially, you just tore them out and messed them up. So yeah, it causes big problems when it comes to these mutations. So, substitutions, not that bad, right? Substitutions, you're substituting one for the next. In a silent mutation, say you go from an A to a C, but it doesn't change your amino acid. AAA codes for leucine, AAC codes for leucine. Nothing happened, right? Same amino acid was your product. That's known as silent, doesn't affect it. It was a mutation, yes, but your amino acid was the same, so your protein's the same, no problem. A missense mutation basically swaps one amino acid for another. So you were supposed to have methionine, but you got arginine. Okay, still not that bad because it's only one of those amino acids is wrong in your big, big, long chain of amino acids. So it's not that bad. A nonsense mutation makes no sense. You were building a protein and all of a sudden, you reach a stop codon. And you're like, dude, I just started. So it's an incomplete protein. Right? It doesn't actually get to where it's supposed to go. It doesn't get to the finished product. You stop it early. Okay? It's missense. It doesn't make any sense. Now, when it comes to insertions and deletions, this is the bad one, right? This is where it actually really, really affects you. So these guys affect the entire triplet and the entire codon, which affects your amino acid, which affects your reading frame. So it affects everything. So these guys right here, the substitutions and the deletions, these are the ones that are really, really bad. Now, You will have to do this for homework. You will have to do this on an exam, pending I can actually make this happen. But we're also going to practice it here in class. So what I want you guys to do is I do actually want you guys to take notes on this, and I want you guys to show me, snap a little photo of it, type it into your computer. I'm going to place a spot online for you guys to actually submit this. This is considered classwork. What? At home? Yes, I know. All right, so here's how you read this. Remember, your codons come in threes. So let's start with the start codon because that's the one that everybody needs to know. Remember, nothing can happen until you get to that start codon. So we're looking for the AUG, right? That's our start codon. So let's see what amino acid AUG codes for. So first up, we have our first codon right here on the left-hand side. It says first codon. You have U, C, A, and G. We're looking for A, okay? A. And you can see AUU, AUC, AUA, AUG. Cool. ACU, ACC, ACA, AC. So now you look at the second part, which is going to be the top part here. So it says the second base pair. U, C, A, and G. So we're looking for AUG. We go to A, we go to U, so we're in this column right here for all the U's, and then we just find it, because the third column is on the right-hand side, but I always just find it easier just to find it. AUU, AUC, AUA, oh, AUG, found it. So it codes for methionine or the start codon. Okay, so when I want you guys to write this out, you can either write out methionine or MET, or you can write out start. Either one is correct, but you have to start with methionine or start. You can't start with proline. That doesn't make sense because it's not the start codon. Okay, so let's do another one. I want you guys to practice this. So let's do GCA. Okay, so the first thing we want to do, we want to find our G. We're down here. We want to find our C, so it's going to be the second column. It's going to be this guy right here. So we go from G. Second column, we have GCU, GCC, GCA. Okay, now we found our GCA. GCA codes for alanine, also abbreviated ALA. If you just write ALA, that's okay, I know what you mean. Okay, this is alanine. We found our codon. So first we had methionine, second we have alanine, and so forth and so on. So if this seems a little bit confusing to you guys, practice it. Just make up 3 letters and give yourselves a bunch of codons and then go find them in this chart because you will have to do this at home. In fact, we're also going to do another part of in-class work right now, which you guys are also going to submit to me via Canvas. So if you have questions on that, go ahead and email me. Otherwise, there will be a spot under assignments. I'm going to give you guys probably 4 or 5 days to do this. It's really easy. You could actually probably do this while I'm making this video right now. So let's go to the in-class group work. Okay, so let's start with our in-class group work right here. I'm going to walk you guys through this. If you can't see it, hopefully you can zoom in. I'm going to try to zoom in once I'm doing the editing too as well. All right, so we have our... Okay, so we have our very long sequence of DNA first. This would be kind of the same kind of thing I'm going to write on your test. So we have our DNA. In this case, we're reading it from the 3' to the 5' end. So the 3' end goes here. The 5' end is down at the end. I'm going to read this out loud so you guys can make sure to copy this down. G, C, G, T, A, C, C, G, A, T, 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 A, C, G, A, T, C, G, G, C. 5'. Prime is just a little notch, kind of looks like an apostrophe. Okay, so that was my DNA sequence. That will be something that I'm giving to you guys. Now you guys are actually going to have to go and code this into RNA. Remember, A goes with U, C goes with G. If you are confused about the TC thing, just write it out. In fact, that's what I'm going to do right now, show you guys how to do it. I'm going to write it out as if it were DNA, and then I'm going to switch it to RNA real quick. So remember that these are complementary angles. They're going in opposite directions. So if I'm going from 3 to 5 for my DNA, for my RNA, I'm supposed to go from 5 to 3. Okay, 5' to 3'. All right, so let's get that started right now. So we had G, which means we have a C, C, which means we have a G, G goes to C, I'm not going to keep reading this, T goes to A goes to T for now, and then I'm going to do the rest of them. prime at the end down here. Now, remember, we're an RNA, so there are no T's, but this is the easiest way to do it to not confuse you guys. So now let's erase all these T's and just swap them out for use. Okay? Okay, hopefully you guys have done this right. If not, the correct sequence is CGC, AUG, GCU, AAA, UGC, UAG, CCG. Okay, I know it sounds like a lot, but it's really not. Now, let's start codons, okay? Very simple. Remember, codons are just a sequence of 3, okay? So our first sequence of 3 is going to be this guy right here. Okay, so we're going to write C, G, C. That's our first codon. Here's our next one, A, U, G. That guy's familiar, right? G, C, U, A, A, U, G, C, U, A, G, 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 C. Okay, these are all of our codons. Remember, sequences of 3. Based on these codons, now we're going to read that chart, and you're going to tell me what proteins go here. Or sorry, what amino acids go here to build our protein. I will give you guys a hint, okay? Do we start with CGC? Why not? Because AUG is the start codon, which means when we're reading this, nothing happens, nothing happens, nothing happens. AUG, now we can start. So my first amino acid in this sequence would be methionine. Okay, not whatever this code's for. That one doesn't count because we hadn't hit our start codon yet. Remember, you can also write, start, methionine, you can write either or. Both of them are totally acceptable, it's fine. But remember, this is the first one. So next we would read this one and this one and this one and this one, and if we find a stop codon, we stop. Okay, so you never start until you get the start codon, and you never stop until you get the stop codon. If you do get the stop codon, do not write stop and then arginine or whatever it is, right? You stop, right? This would just be blank, and you would stop right there. So that's what I want you guys to do for homework. If you think you've done it incorrect and you want me to check before you submit it, just go ahead and email it to me. Otherwise, practice this. This is going to be something that you are going to have to do for your test, and it's really not that hard. So make sure that you guys get that practice in, and if you have any questions, you can come see me as well. Okay, so hopefully you guys got this chart down. Hopefully you guys got the in-class work that we've been doing, and that you really, really do understand this, because it will be, again, upcoming on your test, homework assignments, as well as potentially short answer. Just making me a protein kind of thing like that. It's actually pretty easy, but you must practice this. A lot of times people are like, oh yeah, I totally get this, and they totally mess it up, especially going from the A to the T's and the U's and all that. Very confusing. So do what I did. Go from the A to the T, right? The T to the A, but then erase all the T's if it's in the RNA sequence. Don't erase the A's. You're not going to even find a start codon then. You probably did something wrong. If I've given you something and you guys don't find the start codon in that sequence, you've probably done something wrong. So double check on that while you guys are practicing. Okay. So where do these mutations come from? Remember, I told you guys that some of them are good. Some of them allow genetic variation in the population, which is really a great thing. Remember when we were talking about the giraffe's necks being slightly larger? That was a genetic mutation, right? Through some mutation, he just got a little bit taller and a little bit taller. Maybe he had the protein for a taller neck. Now all of a sudden the giraffes with taller necks are doing better in the population. They're able to eat more because they can reach the trees that nobody else can. That means they're nice and healthy. That means they can have all the babies, right? That is a good thing. That's a good mutation. Sickle cell, not so much, but again, you're resistant to malaria, so not really a bad mutation if you live in areas with high, malaria rates. I'm just joking on myself. Excuse me, okay. All right, so remember, some mutations are good, some mutations are bad. The whole point mutation with the malaria, not necessarily great. Sorry. Delete all of this. X. Okay. So just remember that not all mutations are bad. Some mutations give us genetic variation, which is really, really good. Some, cause bad things like sickle cell, but kind of have a good thing tied into that, meaning you are resistant to malaria. So not necessarily a bad thing, especially if you live in a very high malaria risk areas. So sometimes they can be harmful, sometimes they can be not harmful, and sometimes they come totally random. So sometimes they're kind of at the you're just going through the DNA replication and you just happen to put the wrong nucleotide in. So that's a random mutation. Sometimes it can be caused by external factors as well. Sunlight, UV, can actually cause mutations. So too much exposure to direct UV light can actually go in, penetrate your skin, and mess with your DNA. So too much exposure to things like UV light, like the sun, can actually cause DNA mutation. So this is kind of a big deal. Smoking, alcohol, all of these things can actually lead to mutations. Mutations in your genes that you will then pass on to your kids. These things you don't really think about when you're sitting there having a glass of wine during this, lockdown. But sometimes, especially in heavy doses, obviously if you're having a single glass of wine, it's not going to cause a mutation. But if you are, a heavy drinker, an alcoholic, or you smoke cigarettes a lot, all of those things can lead to mutations. Those are what are called environmental factors. They can affect your genes. They can affect your DNA. It's kind of scary, actually, how fragile we really can be sometimes. So with that, I leave you with a terrible dad joke. Get it? Rearranging DNA, just like you guys missed so much from class. But thank you so much for sticking with me. I know this is not easy, but thank you guys again, and I will see you soon.